behavior, genetics, health, and aging. Bob has won numerous awards, too many for me to list, because it would take up too much of our time and we wouldn't get to his talk. He's been named one of the top 100 most impactful psychology researchers in the world, according to research.com, which is pretty cool. And Bob is going to talk today about his work, Nature or Nurture or Both, how genetics and experience impact mental health. And one little note before we start is just for the CME credits, I'll put the code in the chat here. The code is 2077, and we'll repeat that a couple of times for everybody.
Okay.
So without further ado, I think we'll get started. Thanks, Bob.
Thanks, Michael Paul. I appreciate the invitation. Nice to see everybody in person in this semi-post-pandemic era we live in, or whatever we're calling it these days. And I'm sure there are a bunch of people online too. So thank you for making the time to attend today. I'm going to talk about the kind of work that we do at the intersection of nature and nurture, which is something I've been, fascinated with throughout my whole career. The kind of question that's always fascinated me, really since being an undergraduate and first learning about, human individual differences, is how do we understand that intersection, nature and nurture? It seems a priori important to think that both are likely to matter, but like with many things, the devil's in the detail, right? We can assert that both are likely to be relevant, but how do we get kind of a scientific understanding of their direct interplay? So the talk that I put together for today talks about some behavior genetic concepts, strategies, and designs. So the kind of work that we do these days, on human individual differences, particularly as relevant to mental health and psychopathology and that kind of thing. And I'll present a recent example of gene by environment interaction, or what's called, biometrical moderation, some of our twin research, and also a brief overview of some of the contemporary molecular genetics of DNA-based research that we do in my research group. So start with a kind of basic textbook introduction to the idea of twin studies. As many people, I'm sure, already realize, but it's worth reviewing because it's so central to the way this kind of research works. There's two kinds of twins, right? Identical twins and fraternal twins. And it's a fascinating experiment of nature that twins even exist. And they really, these two types of twins are really typological because they derive from very distinctive biological processes. So identical twins, a single egg fertilized by a single sperm that then splits in two. So you get people who share all their genes, roughly speaking, right? So it would be identical by descent, right? If you want to use technical terms for gene sharing, but basically at the nucleotide level, it's the same nucleotide sequence in these people, or should be throughout all, sort of somatic cells. Fraternal twins are like siblings that happen to have been conceived at the same time. So the mechanism there is different, right? This is separate eggs fertilized by separate sperm, and so you get people who are the same genetically as siblings, but happen to have been born and conceived at essentially the same time, because there are two eggs that are fertilized by two separate sperm. Here's another illustration of this basic, sort of biology, right? So identical twin, one egg, fertilized egg, zygote splits into two, creating, people who are the same genetically, versus fraternal twins, where two separate eggs, two separate sperm. And so because of that separation, you can get same or opposite sex. Fraternal twins, identical twins are always the same sex. So why is that relevant to, trying to understand nature and nurture issues in mental health? Well, this all relates to the degree of genetic resemblance among different kinds of relatives and how you can leverage that information to make some inferences about nature and nurture kinds of issues. So this again just illustrates identical twins are 100% similar, fraternal twins 50% similar on average, second degree relatives like a grandparent or an uncle 25% similar, and so on and so forth, first cousins, second cousins, and unrelated. And you could in principle study all these different kinds of relationships, but the most sort of informative relationships are really the twins, because there you have all twins are matched for age and could be matched for sex. Plus, the identical twins are matched for their sort of underlying nucleotide sequence, which is different, right, from the degree of matching for the, fraternal twins who are 50% similar on average. So it's that contrast that gives you kind of a handle on nature-nurture issues. So that leads to some key concepts that fall under the rubric of what is called ACE decomposition in this literature. And you'll see that A, C, and E refer to components of variation in humans. So that could be individual differences in, risk for a mental disorder or the actual prevalence of a mental disorder. So twin research participants provide one way to really integrate our understanding of nature and nurture. Again, MZ twins share their DNA nucleotide sequence, and fraternal or dizygotic twins share on average half their polymorphisms, or different forms of genes, so different single nucleotide polymorphisms across the entire genome. And because of that difference, that allows you the leverage to infer some effects if you're studying variation in something that you're interested in, again, like a mental disorder in groups of twins. So the first effect I want to talk about within the ACE model decomposition is additive genetic effects. So conceptually, this is the observed similarity between relatives that's attributable to their relationship, taking into account environmental effects. So you'll see that the way that ACE decomposition works is that all the effects are net of each other, right? So basically you're controlling for the environmental effects in examining the genetic effects and vice versa. The second component is shared environmental effects. So that's the similarity between relatives that's attributable to being raised in the same household, essentially, right? So it's how similar people are, net of genetic effects, because they were raised together, right? And C is conceptually independent from A, right? So that's the beauty of these sort of twin study designs, is that you can differentiate genetic effects sitting in the A component from common environmental effects, which are sitting in the C component. And there's a final component, which is non-shared environmental effects. So the environment that makes people different in spite of the presence of A and C effects. So you can think of that as the dissimilarity between relatives taking into account A and C. So these are conceptually different kinds of environmental effects that the twin design allows you to sort of pull apart or disambiguate, right? So with C, it's something that's making people similar, again, controlling for genetic effects within families. So that might be things like the literal household that you grew up in with your siblings, or family level religious traditions, right? Something that's in the environmental circumstances that might be considered relatively independent of genetic effects that's making people similar by virtue of being raised in the same household. And that's different from non-shared environmental effects, which are things that are causing people to be different from each other, in spite of the fact that they share rearing environments and a certain amount of genomic material. So a good example might be unpredictable accidents, right? If you happen to have an identical twin and one person is unfortunate enough to have a serious accident, think of like a motorcycle crash or something else that, damages the prefrontal cortex or something, right? You're going to get different phenotypes in spite of the fact that these two people share the same underlying genetics. Because of this, profound environmental insult to the brain, right? And that's going to show up like non-shared environmental effects. If it's something that happens to a person, randomly and stochastically in a way that, might be considered conceptually independent of their genotype. Okay, so those are the A, C, and E concepts that we're going to use, as we try to understand, how twins can be leveraged as a source of information about variation. So let me walk through some of how the actual modeling works. This gentleman here is Andre Louis Koleski. Those of you who have some, background in mathematics might remember who he is, right? He was famous posthumously for developing some of the mathematics of decomposition that we're going to use here for decomposing variation in something we're interested in, like individual differences in mental disorder, into these A, C, and E components. So in an A, C, and E model, right, or an ACE model, we have the possibility to understand the relationship between two traits, as well as the variation in a single trait. So what I've sort of portrayed here is two different traits, right? So that could be something like a particular mental disorder and a putatively different mental disorder where we're trying to understand, say, the comorbidity between them, right? That's a way in which these models are frequently used. So we could say something like, in twins, we've studied individual differences in, major depression. We've also studied individual differences in generalized anxiety disorder, and we want to understand the sources of overlap between these phenotypes. So that's one way in which this kind of thing has been used a lot in the literature. Because what we're able to do is figure out A, C, and E effects in common between the two traits or phenotypes, right? Those are the ACE effects on the left. You can see that they're pointed at the first phenotype. Think about that as like major depression. And the second phenotype, you could think about that as some anxiety syndrome. And we want to see how much of the A, C, and E effects are common to the two syndromes. And the phenotype on the right, the second one has A, C, and E effects that are independent of the first phenotype. So kind of residual genetic and environmental effects on the second thing, net of the first one. And to lay out the whole model, we have to take into account that we have two twins, because that's how we leverage the information in the twin data set to differentiate A, C, and E. So we have here both traits. Again, think of it as maybe like two mental disorders we want to study in concert to figure out comorbidity, That could be depression and anxiety. And we have them measured in the first half of the twin pairs. That could be like the people who were born first and the people who were born second within the pairs, or it could be randomized as to who's the first and who's the second twin. And we have those effects for both twins. And then to formalize the model or to lay it all out, we have to add the expectations for the similarity among the twins based on the definition of these A, C, and E effects. And that's what these arrows are at the top. So these are correlations within the model that are fixed structurally. So what they are basically is defining the effects, The part that where you're trying to figure out the magnitude of the effects are the arrows coming from the A, C, and E parts pointing at the traits, Those arrows that don't have anything on them. And the definitional part of the model is at the top, So it's that top part that's defining what we mean when we say A, C, and E, like those conceptual ideas now defined mathematically. So A effects are 1 for identical pairs because they share the same underlying, again, nucleotide sequence. And those effects are 0.5 for dizygotic pairs because on average, allelic sharing, across pairs of siblings, which is what DZ pairs are, they just again happen to have been conceived at the same time, is a half on average, There's a 0.5 probability that any particular allele is going to be shared between those twins at any particular point on the genome that happens to be polymorphic. So that's the definition of A effects, It's how similar people are based on their degree of genetic similarity. Now, C effects show a correlation of 1 from twin 1 to twin 2, because definitionally that's what they are, These are things that make people the same, basically, because they grew up in the same family, So to the extent that we define C effects that way at the top, what we're deriving at the bottom is the degree of influence coming from C on the phenotypes that we're trying to understand. And conceptually, E effects are things that make people different in spite of the fact that they share genetic material and grew up together. So you can see that E effects are defined by no connection between the two twins, because remember, that's the thing that's kind of random or stochastic between people within families, Again, remember my example, something random happens to somebody and it doesn't happen to their co-twin, like an accident or something. Well, that's conceptually what E is, and that's why it's defined this way in this kind of model. Okay. So I'll give you guys an example of some research, that takes this kind of design into account. That's pertinent to, things that would be of interest in psychiatry. This is childhood externalizing problems and marital discord. So these are data that come from the Minnesota Twin Family Study, So I'm part of a research group on campus called the Minnesota Center for Twin and Family Research. And one of the cohorts that we've studied for, decades now is called the Minnesota Twin Family Study. We began studying these twins when they were 11 years old. Pretty big sample of twins, about 750 pairs. We don't study opposite sex DZ pairs, so we just have same sex, fraternal pairs in this sample, but we have different pairs that differ in biological sex, as well as zygosity. And the phenotype we're going to focus on here that's, again, of relevance to, psychiatry is childhood externalizing problems. So these are things that in the DSM nosology would be called conduct disorder and oppositional defiant disorder. And these were reported on by mothers using a standardized diagnostic interview, that we had our research assistants administer. Also indicators of self-reported delinquent behavior and information from teachers. So kind of a pretty thorough attempt to characterize externalizing in these kids at this age. Now, what we're going to do in this model takes that ACE model framework and takes it a step further because it adds the possibility of a moderator. And I'll show you the diagram in just a moment here. But this is something that I think is really important and fascinating, right? So instead of saying that the ACE effects exist in a constant way for the whole population, when we introduce the possibility of a moderator, we're looking at gene by environment interaction kinds of effects. Because what we're going to say here is that the magnitude of genetic and environmental influence on externalizing might be a function of parent relationship quality. So hopefully that idea is sort of clear, like conceptually and perhaps even clinically, right? So we're interested in the etiology, essentially, right, of externalizing problems in childhood. Like, where do they come from? Are they genetic? Are they environmental? And what we're saying here is, well, the degree of genetic and environmental influence might be a function of something about the family environment. In particular, the degree of stress within the parental relationship. And we measured that by using something called the dyadic adjustment scale. So this is basically, are the parents, getting along with each other? They have a loving relationship, or are they really fighting constantly, very unhappy, probably considering separation or divorce, that kind of thing. So that's what the DAS scale is getting at. So the parents were asked, like, how's it going in your relationship, right? Things going well, you're on the same page as your spouse, and it's all good, or there's a lot of conflict in the family. So the idea here is that the degree of family conflict might affect the etiology, essentially, of childhood externalizing problems by shifting the degree of genetic and environmental influence. And we want to be able to study that in a formal way, using this research design. And just a little bit of kind of like, the underlying statistical model that allows us to do this. So recall in the previous diagram, we had those ACE effects, A, C, and E, pointing at the thing that we were interested in studying. So the phenotype here, like the thing we're studying, childhood externalizing behavior. So when you see phenotype, think this is kids who are acting out versus kids who are not acting out, basically, right? So conduct disorder and oppositional defiant and stuff like that, to use, psychiatric terminology. And we're going to look at A, C, and E effects on externalizing that are unique to externalizing, right? That's the stuff that's just pointing at the phenotype, and those that are affected by the moderator. So remember, the moderator in this case is, are the parents in the family getting along with each other, right? Is it a sort of positive family environment, or is it a conflictual family environment? And we have the A, C, and E effects just as before. But what you'll see on the paths, right, so those lines connecting the A, C, and E effects with the moderator and the phenotype is a little more algebra. So we have this, A part, for example, plus the beta times the moderator. So what that beta times the moderator part is doing is it's saying the A, C, and E effects might be a function of the moderator level, right? So recall again that the moderator is basically, are the parents getting along with each other, or are they fighting a lot? So what this model is saying is, well, maybe the A, C, and E effects on this thing we're interested in, childhood acting out behavior, might change as a function of the level, right, of the moderator, which in this case is parents getting along or not. So this model allows us to say, basically, is there evidence for a gene-environment interaction, right? Is it the case that the degree of genetic and environmental influence on kids externalizing is potentially a function of whether parents are getting along or not, empirically? Like, we're going to use data to answer that question to see the extent to which, parental relationship quality is moderating or changing the degree of genetic and environmental influence on kids acting out. And this model allows us to do that because it allows us to estimate those beta parameters, essentially, right? The extent to which the moderator is changing the A, C, and E impact on kids acting out. And that's what we found in this study, that there is evidence for moderation. So at the top is the A, C, and E model not allowing for moderation. So just give you an idea of kind of what the literature might look like about the etiology of externalizing before you take moderation into account. So across the x-axis of these figures is parental marital discord. And across the y-axis is the proportion of variance on the left side and the amount of variance on the right side. So the proportion just takes A, C, and E, scales it to 100 and divides it, so it's a percentage. The unstandardized estimates are on the right, so just total amount of A, C, and E. They're similar in this case in terms of how they work. But the important thing to pay attention to is at the top, we're not taking the moderator into account, right? We're saying genetic and environmental effects on externalizing are constant, regardless of what's going on in the family. So kind of a traditional account of the etiology of childhood externalizing. And what that says, right, is that, well, we have evidence here that genetic effects are important for childhood externalizing. I see the A effects are sitting right around 0.4-ish, so about 40% of the variation in childhood externalizing is attributable to genetic effects per this model. And then there's also, common environmental effects at around the same level and some unique environmental effects, right? So the traditional model says, yeah, well, childhood externalizing is a function of both genetic risk and also environmental circumstances that people share, and to a certain extent that people don't share. But those environmental effects are anonymous in that top model. Like, we don't really know what they are. We infer their existence from the patterns of family resemblance. But what the bottom part does is it says, okay, let's take a look at the extent to which the relationship quality of the parents might be changing or affecting the etiology, basically sources of genetic and environmental influence on kids acting out behavior. And that's what we found evidence for here. So to interpret the bottom part, remember the x-axis is the degree of parental discord. So further to the right is more parental discord. Further to the left is less parental discord. So what you see if you want to interpret the bottom ones is that high levels of parental discord, genetic effects are suppressed, right? That black line is lower. See, those A effects are smaller to the right side of those figures. And environmental effects are enhanced under conditions of greater parental marital discord, like further to the right on those graphs. Further to the left is parents who are getting along, right? And what you see there is that genetic influences are bigger than, environmental influence. So conceptually and clinically, what do we have evidence for here? When parents are fighting, that environmental etiology shows up in the etiology of the externalizing presentation, if that makes sense, right? So when parents are fighting, it changes, it shifts the etiology to be more about factors in the family. Right? You can see that at the bottom, right? In conditions where parents are having a rough time, they're fighting, the family environment is very negative. The causes of individual differences in kids acting out is more environmental, coming from, right, that, deleterious, problematic family environment. To the left, though, individual differences in kids acting out in families where parents are getting along is more genetic, which makes, I think, also conceptual and clinical sense, right? Like kids who are acting out, in spite of being in a loving, caring family relationship, presumably have a high genomic risk, right, for acting out kinds of behaviors. So that's what this provides evidence for, basically, right? That there's not one constant etiology of childhood externalizing behavior, but rather that etiology is dynamic, right? It's based on things that are going on in the family. And here, by using this kind of moderated twin design, we can present some evidence for something a little bit more dynamic about, the etiology essentially of child externalizing. Okay. So then I can say a little bit about our research with DNA, right? So in this kind of work, we're interested not just in, using twin and adoption designs to derive inferences like I was just talking about, but we're also interested in working directly with molecular genetic data to ask similar kinds of questions about the integration of nature and nurture. So just a little bit about kind of the history of this stuff, especially as it applies to the psychiatric literature. Starting in kind of the late 90s, early 2000s, there was a really big interest in candidate genes in this literature. And so that was the idea that we could genotype specific polymorphisms that were of putative biological relevance, right? So for instance, people were interested in studying polymorphisms that were relevant to monoamine neurotransmitters like, dopamine and serotonin. And there was a large literature on things like those variants and how they might relate to psychiatric phenotypes. And in general, that hasn't worked out very well, right? So there's a lot of interest in that, a lot of research done on that, a lot of problems replicating those effects, right? So people would look at things like, the serotonin transporter variant, was that relevant to risk for depression? Different papers would find different things and generally not replicable. So then one of the next things that was attempted in this literature is candidate genes as moderated by the environment. So there were a series of very influential papers on things like, okay, well, maybe the serotonin transporter is relevant to depression, but only in people who have experienced psychosocial stress, which makes some intuitive sense and is a similar idea conceptually to what I was just describing from the twin literature. Unfortunately, that approach also hasn't worked out very well and tends not to produce replicable effects. And so you can ask yourself, well, why has that been so difficult, right? Why is that research not producing stuff that's replicable? Well, I think one of the reasons is probably that we may not have identified the relevant genomic variation. Like basically the whole idea of looking at specific candidate polymorphisms is not the most effective way to characterize all the information in the human genome. So as you'll see in the next upcoming slides, there's much more information in a human genome than just these very single, very narrow, very specific polymorphisms, even though they might be of some functional relevance, right? There's just far more genomic variation out there that isn't being characterized by this candidate kind of approach. So genomic variation, is unlikely to be well characterized by single variants. And also the environment might not be completely exogenous to the organism, if you will, right? So in this kind of gene by environment approach with candidate genes, it's like the environment happens to people in a way that doesn't take into account correlations essentially between genes and environment, right? So that gets interesting to think about. Do we encounter environments randomly? Well, probably not, right? Like think about all the pathways that led you here today, right? They're not independent of your genotype, most likely. See what I mean? Like you're not here for random reasons, right? You're here because you're interested in psychiatry, you've studied in the field, you have some professional role in this. And some of that is being driven by, genomics, right? So you're not encountering this environment in a totally random way. You're encountering it in a way that's correlated with, all the things that have happened to you in your life that have something to do with genetic variation, if that makes sense, right? So genes and environments are probably more correlated than we, thought in this kind of candidate gene by environment approach. So a lot of the research now in psychiatric genetics, focuses on genome-wide association, and that's a technology and kind of study design that was made possible because of the Human Genome Project. So in this approach, what you're doing is scanning the entire genome for potentially relevant single nucleotide polymorphisms. So recall that, genomic variation is made up of these nucleotides, G, A, T, and C, right? And so I'll show you in the next slide kind of like how this works across the genome. But it is the case that we're all similar to each other on most of those nucleotides, but every 100 or so base pairs, you're different from me and you're different from somebody else in this group, right? It's that variation that we can now actually characterize, which is really kind of remarkable technology, right? So by taking a sample of, cells, we can, go in and have a look at genomic variation and figure out your particular pattern, right, on these single nucleotide polymorphisms and how that's different from somebody else's, giving us a lot of leverage to study this stuff scientifically. So I'll give you an example of this kind of work. This is from the Social Science Genetic Association Consortium. So we did a project, fairly recently focused on depression and neuroticism and subjective well-being. So again, an example is pretty relevant to psychiatry, right? We were trying to figure out, could we hunt for genes essentially that are relevant to depression and other nearby phenotypes, like a person's sense of well-being and their neuroticism, which is a personality trait that's clearly relevant to, risk for ultimately developing depression. And we were trying to illustrate how these things kind of work potentially in concert. At the time we did this, these were pretty big samples. Samples are getting bigger all the time. So in this literature, we're now up to millions of observations, but this example involves hundreds of thousands of observations. And why do we need such big samples? Well, you'll see in a moment that that's part of the key to what is now happening in human genetics. Basically, each of these individual SNP effects is pretty small by itself, but in the aggregate, they seem to be relevant, right? So it's all the information essentially in your genome, not this particular SNP or gene or this particular gene here, but like all that information that's contained in your genome that's relevant to risk for developing, say, psychiatric illness and so on. So how does this genome-wide association study approach work? Well, we genotype millions of SNPs across the genome. Again, those are individual nucleotide bases of about 3 billion total, right? So that's your whole genome, about 3 billion of these, nucleotides. And we're genotyping the ones where variation is frequent enough to have some leverage to study it. So typically that's called the common variant kind of approach in this literature. So that's where the minor allele or the less common allele is at least present in 1% of the people. Because it's interesting to think about, like, there are reasons to study even rarer variants, but then that diminishes your ability to study it because only a few people in the sample are going to have that unusual variant. But that may be important, right? Rare variants might be particularly important to psychiatric illness. So that remains sort of an interesting thing to think about in the literature. So anyway, 1 in about every 300 or so of these base pairs is different between, again, you, me, somebody else in the room, any other human being, right? So as you're going along the genome, right, here's a cartoon illustration of the genome, right? You have the same sequence as another person most of the time, right? 299 out of like 300 times, but then every once in a while there's a switch, right? There's a difference between you and somebody else. And that's called a SNP, or a single nucleotide polymorphism. So in the example here, right, the person at the top has a different genomic variant from the person at the bottom. But on the other points along the genome, we have the same variants. And again, that happens about every 300 or so base pairs. And in something that I was just working on like a couple weeks ago, a Genomewide Association study of personality, actually, we looked at our data and figured out that about 16.7 million SNPs were available in every person. And that was after imputing the whole genome. I'm not going to say too much about how that works, but basically, using contemporary molecular technology and statistical approaches, we can kind of figure out the whole genome of a person with reasonable accuracy. And the point I'm making here is there's about 16.7 million places on the genome where you're different from me, and you're different from the person you're sitting next to, etc. So you can see the bioinformatic problem we face in this field. Like, there's a lot of data in your genome and a lot of information. And what we're encountering here is, how do we make sense of that, right? If you get a person's, blood cells or saliva or something where you can figure out their genotype, right? You're getting, 16.7 million data points on a person, which is too much information from a statistical standpoint. Similar problems exist in, other kinds of contemporary technologies like imaging and so on, right? There's just a lot of data coming from these things. So that's part of the challenge, I think, is just straight up bioinformatics, right? Like, what are we going to do when we have so much data? We're going to need very big samples to make sense of this, because it's not a few observations on a person, it's millions. Okay. So what did we find in this GWAS study? This is something that's called a Manhattan plot. And so the way this works is the x-axis is the linear genome from the first chromosome to the last one, leaving out the sex chromosomes in this case. So that's just the genome like lined up physically from the beginning to the end. And then on the y-axis are p-values, which are basically how significant was the effect of a particular SNP in this study when we're trying to figure out, does that locus, right, or that particular SNP at that location have something to do with, in this case, subjective well-being, depression, and neuroticism. So we found some, significant hits here, right, across the whole genome. Some evidence that some of these are unlikely to have occurred by chance because their p-values are so small, right? So a good chance that some of those specific SNPs are relevant to these phenotypes. Those are characterized by those RS numbers that are floating above the significance level. So some of that was going on, but the part that I found, particularly interesting in this study is this part about basically like comorbidity, like trying to figure out, are these really separate, phenotypes or conditions, or do they share some etiology at the genomic level? So we looked at that here. At the top, we see what's called the genetic correlation. So that's basically, were the SNPs that were found to be relevant to one phenotype correlated, or basically the same SNPs as those that were relevant to some other phenotype. So we found here that subjective well-being and depression are pretty highly correlated. Well, they're negative in this case because well-being is the opposite of depression, but basically a strong overlap in the relevant SNPs. Same thing for well-being and neuroticism, and same thing for depressive symptoms and the personality trait of neuroticism. So some evidence here that the genomic signature, if you will, sort of the genetic aspect of the etiology of these things is largely in common at the molecular level, right? So like being a neurotic person with that kind of personality style is picking up on largely the same genetic variation as formal depression kinds of, assessments essentially. And just to show that it's not something, that makes little biological sense, right? We threw in height, right? Like how tall are you, right? So is the genomic signature of height correlated with the genomic signature of, well-being and depression? Well, the answer is no. And so it's those X's on the right side on the top, right? Small genetic correlations between how tall you are in a genetic sense and your propensity for depression in the genetic sense. And then on the bottom, we're looking at like other studies that are not this study and what they found for SNPs that are relevant to psychiatric phenotypes. So Alzheimer's, formal DSM style anxiety disorders, autism, bipolar, and schizophrenia. And yeah, we found a good deal of genetic overlap with the anxiety disorders that were studied in other studies, but not so much with Alzheimer's, autism, bipolar, and schizophrenia. So that's kind of supports this idea of like, well, depression and anxiety are part of a spectrum of interrelated, conditions that we might call the internalizing spectrum, right? That these are things that are often comorbid, right? We certainly see this clinically, right? Because, well, they share a lot of their genetic etiology. Okay, maybe one more example before we sort of open up to questions. This has to do with experiences of discrimination and the experience of anxiety and trying to take a molecular genetic approach to finding an environmental effect, essentially, of discrimination on anxiety. So the thing that we got interested in here is, well, okay, if we know something about genetic effects on something like anxiety, could we take that into account in searching for a bona fide environmental pathway linking people's experiences of discrimination with their experiences of anxiety? And this is work we did in something called the midlife in the US sample. So, my job in this grant is to help run the Genomics project. This is a large national sample funded by the National Institute on Aging. Participants here are aged 25 to 74, and we were looking at a group of biologically unrelated participants. And we studied, again, discrimination experiences. So that, our measures can be divided into kind of three types of experiences: daily discrimination, like feeling on a daily basis you're treated with less respect than others; lifetime discrimination, over the course of your life, do you feel like you've been discriminated based on various social identities like gender, age, and so on; and job discrimination, so experiences like you feel like you've been unfairly given, jobs that other people didn't want to do. And we're trying to understand the impact of those discrimination experiences on anxiety. So I'll show you some of the anxiety indicators in the next, slide. So this study uses this idea of polygenic risk scores. So I'll just explain a little bit about what those are. That depends on estimates from a genome-wide association study. So here's another one of those Manhattan plots. This is just an example, right? Again, just to remind you what a Manhattan plot is. Remember, in GWAS, we're figuring out across the genome which SNPs might be relevant to something we're interested in studying. We can turn that information into something called a polygenic risk score. So when we're doing this work to discover the relevant SNPs, we estimate associations between the phenotype in each SNP. That's that GWAS study kind of design that I was talking about before. And if we know how much a SNP is relevant to a phenotype, we can compute basically the risk for that phenotype in a separate sample from the discovery sample. So if we know something like what's in that, figure there, like how much these SNPs are, supposed to be relevant to something we're trying to understand, like anxiety, and we take that information and port it over to a new sample that wasn't used to discover the relevant SNPs, we can basically use the genotype to predict a person's risk for something we're trying to study. Right, so this is like using a polygenic score as an index of risk for something like anxiety. So what we're going to do here, right, is use genomic data at the molecular level, similar to how we use the twin study approach, to try to, control for genetic variation when getting at environmental effects. Another interesting thing about polygenic risk scores is they tend to be normally distributed. So they tend to show a kind of Gaussian curve like this. And they use this, they allow you to control basically for genetic effects in looking for, environmental effects. So that's what we did in this paper that appeared recently in the PNAS journal. So again, remember the research question is, can we use discrimination experiences to predict anxiety? And the answer is yes, right? There's a correlation between discrimination and anxiety in this figure. That's that 0.53 right in the middle. So people who feel like they've been more discriminated against, subjective experiences of discrimination is predictive of their level of anxiety. But the piece of this that we did that goes a bit beyond the existing literature is the set of covariates at the top. Maybe I'll start at the bottom with it. What did we mean again by discrimination and anxiety? So both of those variables, discrimination and anxiety, have multiple indicators, like multiple ways we measured them. Remember, again, discrimination involves daily experiences, lifetime experiences, and job discrimination. And anxiety involves, trait anxiety, social anxiety, diagnoses of generalized anxiety disorder, so DSM style phenotype, neuroticism, negative affect, and this PANIS scale, which is a scale for measuring negative affect. So we're looking at, is there an association between discrimination and anxiety, net of these covariates, right? And so the covariates are the things at the top of the figure. And that's things like age and biological sex and level of education. So typical covariates in kind of psychiatric epidemiology. Principal components control for relatedness among the people of a sort of subtle nature. And I won't say much about that right now. And then these polygenic risk scores for anxiety and depression and neuroticism, right? Like some of the stuff we worked with in the previous study. So what are we doing here? We're controlling for people's genetic risk, right, for anxiety, in inferring that discrimination experiences predict anxiety, right? So there's more evidence here from this design that that's an actual environmental effect, right? That there isn't a confound with genetic risk, because it could be the case that genetic risk for anxiety predicts genetic risk for feeling like you've been discriminated against or persecuted. But we're controlling for that possibility here and making a different kind of inference thereby, right? But because we can control or try to control for that genetic risk, we have some evidence that actual experiences of discrimination predict anxiety in an environmentally mediated way. Okay. So I think this is my last slide, some conclusions. Yeah, so this is the kind of stuff that we do, right? In my lab group, we're interested in multiple methodologies that integrate genetic and environmental factors, right? We take family and twin kinds of approaches. We're also using molecular kinds of approaches. I haven't talked about our epigenetic stuff today, but that's an area that we're getting into that we're very excited about. Various designs I think are complementary to something we've found through the years, right? That there's utility to some of the classical family designs, even if we're sort of getting into more of the molecular technologies. And these approaches remain interesting, to me and the folks I work with, because they basically provide, interpretive leverage, let's call it, right? They're research designs that allow you to say more than there's a correlation between two things, right? So going back to your first, stats and methodology course, like correlation isn't causation, but by using these designs, we can get more at that kind of potential causal underlying set of mechanisms as we try to understand, phenotypes that are relevant to mental and physical health. I think that's it. All right. Thanks so much for listening. Appreciate it.
Yeah.
Well, thank you very much, Bob, for the very interesting presentation. I think people can go ahead and put any questions. People who are on Zoom can put questions into the Q&A section. People who are in the room, feel free to raise your hands, and Bob, you can feel free to call on people. Maybe I'll start with a question that I had that you touched on a little bit, which is kind of the role of genes versus environment and their interaction in personality disorders. You talked a little bit about like trait neuroticism and things, and I wonder if you could comment on, in terms of personality disorder, the role, and any of your work in this area, and the role of genes versus the environment.
Sure. Yeah. So that literature, right, on personality disorders is, where to start with a question that's that broad, but maybe the thing to say is that a lot of the work on personality disorders began with kind of the classical personality disorder categories described in earlier DSMs. So for example, the 10 categories that are described in DSM-4, and as like a lot of that literature now is turning toward more of these dimensional conceptualizations and kind of the literature on the organization of human personality to kind of reorganize the way we think about clinical personality disorder phenotypes. So yeah, I think that the short answer to the question is all the literature talking about, human personality is relevant to human personality disorder, because there's lots of evidence that personality disorders aren't distinct from, continuous personality variation, but there's connections between those kinds of things. So maybe that's a short answer, right? All that stuff on personality is relevant to understanding personality disorder. And, there's evidence of things like, okay, that, the genomics of a neurotic kind of personality trait are similar to the genomics of a borderline personality diagnosis because they share a lot of variation, right? So those aren't entirely separable kinds of phenotypes, if you will. Does that kind of get at your question?
Yeah, absolutely. Thank you.
Okay. Yeah. Any other questions?
Have you seen any like clinical applications of this work, or like I know we're doing some other things with genetic testing from like a polyphy, like psychopharmacology, implementing that. Anything like that with this yet?
Yeah, great question, right? So the question is like, could this lead to, direct clinical applications, right? Could psychiatric genetics, yield polygenic risk scores that you might actually be able to use in the clinic? I think the answer is not yet, because the scores aren't potent enough for routine clinical application. And I think that's a big challenge, because some stuff that's much easier to do clinically, right, is pretty potent in terms of conveying the same kind of information that a polygenic risk score would convey. So in a standard, intake interview, we ask about family history. Are we going to get something more out of a polygenic risk score than we get out of family history? I think that's a pretty tall bar, right? Because you'd have to say, okay, there's information in the genome that goes beyond, the person's ability to characterize, their family history of psychiatric illness. So yeah, that's the hope, right? Of course, that eventually we might discover something in the realm of personalized medicine, right? So some set of SNPs that's going to determine like what medication is going to work better than some other medication, and we can use that to tailor our interventions. But that's a bit in the future, I think. So no, I don't think we're there yet, but of course that's the reason we do research, because there's some hope that maybe eventually we might be able to get something that has that degree of clinical utility. Again, I think it's going to be really challenging, and it's hard to get better than family history, is my sense of how that literature is developing. Because that's so easy to ask, and we ask it all the time, and it works. So, yeah.
We have a question. Oh, wait, sorry, go ahead. Is there another question in the room?
No, I was just saying that was a really good question.
We have a question here in Zoom from Sasha Zagla.
Okay.
She says, thank you for the depth and breadth of this talk. I don't want to lose the nuance that you would prioritize. If I'm speaking with a distressed patient, a parent who says, this is my fault, how might you respond to that?
Oh, that's a really great question. Yeah, so there's a long history of trying to figure that out, right? Throughout psychiatry, right? How much is it appropriate to talk about the influence of the family, right, on the presentation? Well, that kind of thing, right, sounds a bit pathological to me, wearing my sort of clinician hat, right? If a parent were literally saying, this is all my fault, quote unquote, you might want to refer them for services, because that, sounds like depressogenic to me, right? Like nothing that occurs in life is 100% our fault, right? Even within the family. And I think it is helpful clinically to point out that there are, notable genetic influences on psychiatric presentations that go beyond the influence of the family. And that was an important corrective in the history of the discipline, right? Going from the era of the schizophrenic mother, right, to now our understanding that, obviously the environment that the kid is in is going to play somewhat of a role. And that's why we have family education and stuff like that, right, clinically, but it's not the whole etiology. So yeah, I mean, I get it clinically that you would want to then talk in a sort of nuanced and helpful way about, yeah, that's not what the science points to, right? That something is entirely the fault of the parents is false, right? That there's a lot of genomic risk that doesn't come directly from, parents, and is subtle in terms of the environmental influences on a kid's behavior. So maybe that helps, right? You could generically point to the science and kind of correct that, pretty problematic belief, which strikes me as kind of a CBT target for the parent, right? The way you described it. So,
I have a question
Yeah.
through the lens of my background in eating disorders research. There's a growing interest in differential susceptibility in the context of eating disorders, and what Boyce and Belsky and others have written about this, orchid versus dandelion question of whether sensory specific sensitivity and certain traits like that, if you really see not just vulnerability but sort of enhanced functioning, if you will, in optimal environments. But do you think that's mapping on to what we're understanding about genetics, that people aren't just vulnerable, but they could actually have sort of certain genotypes make them potentially able to be malleable in a positive way in certain environments?
Yeah, these are great questions, right? So that was a really interesting question. I'll repeat the questions just to make sure everybody's heard them, like online and stuff, right? So the question was about the possibility that genotypes might promote not just risk, right, for like psychiatric outcomes, but flourishing in certain people, right? So that we might also think about the positive side of all of this. I think that's fascinating and important. I think it's pretty challenging to get that work to happen and to get it funded because of the way like NIH works, in my experience, right? The way that I get these grants, right, to do this kind of work is to write about a clinically significant problem that we can study in these, samples from the community. The aims don't say like human flourishing. Like I could try. I mean, I'm not averse to trying it. Like I could write something like that and see if they bite. But I think in general, like NIH is so organized by, clinically relevant disease phenotypes, let's call it, right? That it's possible to throw in some measures of flourishing and positive kinds of qualities, but pretty hard to get that to be the aims. Right, so I think that's really important, and it's a pathway that you could take in sort of in conversation, I think, with program and with funders, right? That like we really should try harder with flourishing and positive phenotypes. And in my experience, like great idea, important question, like hard to get that conversation to lead to actual research funding, right? That's been kind of my experience of it. So I just turn back to like medical and psychiatric things that that we can and get grants to be able to study these samples, which is not free, and it takes a lot of money to do this kind of research, as you guys can imagine, because of the large sample sizes and stuff like that. So
And because treatment's a type of environment, right? So that's the question of how do we optimize our treatment environments interpersonally and other ways?
yeah, no, I love the question and I love the idea. I've certainly thought about this before and had seen reasons to get excited about it, but hard when you get to the point of like your daily life as a researcher being like, yeah, I want to write something that has that flavor, because it's too distant from like the rhetoric that I generally think will work. Like we're going to do something important about a clinically relevant illness, essentially.
Thank you.
Yeah, of course.
So another question here on Zoom from Rochelle Moen. That's a two-part question. Part one, where are we at regarding renaming borderline personality disorder? Which is something that you and I were touching on earlier in our conversation. And then part two, research shows many people, quote, no longer meet criteria for BPD with treatment, learning skills, and trauma work. I was taught once always a personality disorder, yet the patient holds the disorder for life. Do you have any comments on those two?
Yeah, of course. So I should say to everybody, I'll repeat the question. The question was about borderline personality disorder, and whether there's an effort afoot in the field to kind of deal with the name of that construct, or sort of reconceptualize it in various ways. And some people might be wondering why someone would ask me a question like this, because I didn't really talk about borderline or personality disorder in this talk. So there's another part of my academic life that definitely relates to this stuff that has to do with personality disorders. So just by way of explaining my background, I do a lot of work that's relevant to personality and personality disorders. I published a lot in that area. I served on the DSM-5 committee for personality disorders. I developed a thing called the personality inventory for DSM-5 and was the lead author on that. So like another part of my life is very much about personality disorders, even though I didn't talk about it in today's talk, just so people aren't like, well, why are you being asked this sort of question, right? So that's why. And yeah, I try to maintain an interest and involvement in that field. And I would say that in terms of official nosologies, like the ICD and the DSM, there's, I think, little chance that borderline would ever get renamed. And I think the reason for that is that there's just too much literature surrounding that word, right? And there's been, like a lot of work under that rubric, right? That's been helpful in the way the second part of the question implies. So there's just too many people in the field who are interested in that, clinical syndrome under that name. And, one of the more dramatic things was ICD-11's personality disorder model, which maybe people aren't yet familiar with, but ICD-11's personality disorder model looks very much like the alternative model of personality disorders in DSM-5 and not at all like the DSM-4 category model, right? So the international classification of disease personality disorder approach is very dimensional and very, not like the traditional approaches. And in spite of that, at sort of the 11th hour, a group of actually German clinicians got wind of what was happening with ICD and got very, maybe understandably upset that the word borderline was missing from the draft that was going to be published and really agitated for it being included. So the ICD model, even though like forward thinking in this sort of dimensional way, kind of like borderline is grafted on, so to speak, right? Like it's sitting there in the ICD-11 because it was too objectionable to people working clinically in the field to, remove it, even though the chair of that committee, Peter Tyrer, has been very critical of the concept of borderline. So it's kind of like over his objections. He almost sort of quit, but they got it to happen anyway because it was important and they got the, the ear of people who had a lot of impact on ICD-11. So yeah, I don't see it going anywhere is the short answer, because I've seen, political events of this sort, right, where, and it's understandable, right? I would never want to be, in spite of the work I've done on dimensional approaches, characterized as the person who doesn't recognize how much good work has been done under that rubric, right? Recognizing borderline as a serious kind of psychiatric condition, family supports that surround that concept, patient identities that connect with the concept. Like, yeah, we can say like that's a strange word. It has its origins in kind of psychoanalysis in a weird way, right? And right, so the origins of the term are that it's on the border between psychosis and neurosis because these patients supposedly have features of both the kind of those presentations. Like, yeah, I mean, there's lots of things about it that are problematic, but I don't think it's going anywhere because there's just too much good, helpful work, right? That has been connected with that label. And if you call it something else, like, I don't know, dysregulation disorder or whatever, it's just going to confuse everybody. So I don't see the term going anywhere. If that's my prediction, that would be my prediction. So hopefully that addresses the question.
And in terms of the second part of no longer meeting criteria for a personality disorder like BPD.
Yeah, no, I think what's important there is that personality isn't indelible, right? Like unchanging, right? That's a sort of misconception, I think, of the phenomena that have traditionally been characterized as personality disorders in psychiatry, right? There's this idea that, yeah, I've heard this time and time again in clinical settings, right? Like that patient has a personality flavor, as code for like, this person is a real pain to deal with, right? Like, I don't love that because everybody has a personality and there's parts of personality that aren't about interpersonal difficulties, right? So when people say that clinically, I think what they mean is there's interpersonal stuff going on here that makes it difficult to work on, quote unquote, axis one stuff, because I can't get the patient to like cooperate, do homework, show up for sessions, right? There's like interpersonal stuff that's interfering with being able to do the work that I'm trying to do to help the person. So my point is like that's, I think, more what people mean historically, and it's not really what we mean by personality scientifically, right? So yeah, maybe that's the point. Personality can and does change, right? It takes some work, right, to change one's basic dispositions, but there's ways to work on it. There's ways to change it, and there's evidence, from the study of borderline, for example, that a lot of people do shift over time. So I'm thinking of like Mary Zanarini's work, right, about how borderline, is not an indelible diagnosis, right, in people and fades with time. So yeah, hopefully that answers the question. At least it gives you my angle on that literature.
We have another question from Zoom here from Quentin Gabor. We were just discussing the history of genetics research and psychiatry in our PGY4 History of Psychiatry course. Back in 1928, Hans Luxemburger in Kraepelin's lab conducted the first large twin studies in Bavaria and found concordance for schizophrenia. They used this to promote an understanding that there wasn't mothering parenting that caused schizophrenia. This is an important message, but also parenting and nurture does have an impact. This is super nuanced. I wonder if you care to comment on that.
Yeah, well, history is extremely important, right? And I wish I knew more of that older history better, right? I'm certainly aware of who Kraepelin was and the, textbook on psychiatry and this kind of thing. But that level of, understanding of a literature that's that far in the past, I unfortunately lack that degree of scholarly, depth in understanding that history. But I think that's a fascinating comment, right? That, even that early in the emergence of these kinds of approaches, there was an understanding of the kind of nuanced gene by environment perspective that I think is influencing the work that I and other people are trying to do. So it's great to know that's sitting there in a much, sort of earlier, psychiatric literature, essentially. Any other questions? Michael, Paul, are you still there?
I am, yeah. Mhm.
Okay.
Yeah, we don't have any other questions here on Zoom, but if
Okay.
there are any other questions in the room. Well, then maybe we'll thank Bob again once more for his very interesting talk, and appreciate your time and coming to talk to us. Thank you very much, Bob.
Okay.
And just a reminder for everybody about the CME. Actually, sorry. There. I want to make sure that I have that CME code 2077 for other people, anyone who needs it.
So Lisa Anderson and I are
Okay, I'm
scheduling.
gonna shut it down now, if it's okay, Michael, Paul.
Yep, thank you.
Okay, all right, thank you.
Thanks for a great talk. You two, Bob. Fantastic.